So, just to review, which cell type in your body contains the gene that encodes your photoreceptors? Cells in the retina, cells in your liver, cells in your toes, or all cells?
All cells.
Okay. What is the name for the dimmer switch that regulates gene expression? Starts with a P. Okay. And what are the two main steps that lead from gene to protein? So it goes from, DNA to RNA. Gets ferried out of the nucleus into the cytoplasm, and then is finally translated into protein. Okay, true or false? If we insert a human gene into a bacterial cell, the correct protein won't be made because the genetic code is different between bacteria and humans. Is that true or false? You can be wrong, I don't care. True? False? False, have it. Okay. Have bacteria and humans have the same genetic code? All life on Earth that we know of has the exact same genetic code. So it looks like life only arose once, though it could have arose many times, but perhaps those other life forms didn't survive. Or the other hypothesis is that we're surrounded by other life forms and we just don't know it. Okay? Well, actually, we do have bacteria now that make a whole bunch of proteins that we want. Okay. Now, before genetic engineering, we got proteins mainly by getting animals to make proteins. So, we used horse insulin. We would get horse blood. We didn't kill them. We just took some blood out, removed horse insulin, and then the blood went back in, like kind of giving a transfusion. And then we purified the insulin, and that's the insulin that people got. The insulin, horse insulin, is very similar to human insulin. There is only one problem. Some people are allergic to horse insulin, so they couldn't use it. And if they had diabetes where they needed insulin, they were out of luck. Okay. It turns out that now the human insulin is a hormone, it's a protein, and we can take that gene and through molecular techniques actually insert it into a bacteria, and along with a promoter, and the bacteria will then start making insulin for us. And so we can grow huge vats, 100,000 gallons or something like that. And they'll be making insulin, and then we can take that and purify it down to human insulin. So this had a huge effect, and it's not just for insulin, it's for all kinds of things that some people might need because they were born with a deficiency, a genetic deficiency or something like that, where they don't make the right protein. So now, we also use these techniques in a lot of other ways too. We put genes into plants as well. And we put genes into other animals as well for, let's say, some traits that we, let's say, like. So those are called genetically modified organisms. You've probably heard of GMOs. Oh, yeah, okay. So these GMOs, let's say, they're called transgenic. It's because they not only have their own genes, but we've actually inserted a gene into their genome. In the case of the bacteria, we've inserted the transgenic organism as a bacteria. It can be other things as well. We've inserted genes into plants and for certain traits for a bunch of them. Now, and we actually use recombinant DNA technology, or we did, but that's changing fast, and we'll get to that at the end. Okay. So, can you think of anything else that you know about that, like recombinant and transgenic animals or transgenic organisms? Anybody? Okay. So I'll give you a couple of examples. Oh yeah, one thing I do want to mention. Okay, you've all heard, have most of you heard of GMOs? They've gotten a bad rap. It's like a GMO is something that humans kind of created, transgenic. But if you think of it, plant breeders and animal breeders have been doing the same sort of thing for thousands of years through artificial selection and breeding for traits that they like, right? So if you think about corn, the original plant is called teosinte. We'll look at this in a second, but it had very, very small kernels. It was hardly anything, and yet it was then bred by let's see, Native Americans in the Mexican highlands, and about 10,000 years ago they have the first evidence of it. And they bred for big kernels. Now, if they had the technology that we had, they would have put in the gene that actually makes a protein that makes big kernel corn. But they didn't have that. So it took them a very, very long time to get that. And it also turns out it makes traits, it drags a whole bunch of other traits, because a lot of traits are correlated. So you turn one on, you turn a whole bunch of them on. And a good example of that is there's this researcher in Russia. First, well, they were trying to start, there's a fox there called the silver fox. And so they did selective breeding on this fox. Foxes are wild, and they wanted to breed an animal that was not so crazy and hated humans. And so they bred for a docile fox. And they achieved it. And as the fox, the docile one actually looks a lot like a dog. It's got floppy ears, it's got a curled tail, it's got a personality like a dog and not like a wolf, okay? So you bred for a trait of being, you know, more calm and tame, and all these other things went along with it. So that was the problem with selective breeding. You just couldn't isolate one trait. So GMOs allow you to do that. Okay. So antithrombin is a protein that prevents blood clots. We all have it. We have a gene that makes it. And it's expressed in cells in the liver, which releases an antithrombin into the bloodstream. And that way we don't clot all the time. Because we have a bunch of other things like platelets, which are other proteins that are part of clotting, and then there's a clotting factor, a number of those. Okay. Now, there are some people that are actually born the genetic disease where they don't produce the correct antithrombin, or they don't produce it at all. So they have a mutation. Obviously, those individuals, they have what's called hemophilia, one of the various hemophilias that are out there. So it would be nice if, well, it would be nice if we could actually do gene therapy, which we might be able to do in the future, but for the time being now, perhaps we can get a source of antithrombin and in the human form. So they need 2 copies of the bad gene. So remember, we talked about hemoglobin and sickle cell anemia, and we'll say I just want to make sure you understand HA and HS. Okay, and a person can either be HA or HAHS or HSHS. This is just so you remember what a homozygote is and a heterozygote. So when you have, like this is a homozygote for the normal type of hemoglobin. Same thing with antithrombin, okay? This is the heterozygote, HAHS, where you have 1 copy of normal and 1 copy of the mutated gene. And then this is the one individuals would get sickle cell anemia. So there's 2 homozygotes, that's HA and HS, and then there's 1 heterozygote, HAHS. Okay, now the same thing is going on with the antithrombin. It's a recessive trait. Recessive means that it shows up only in the homozygote recessive form when you have 2 copies of the bad gene. 1 copy, and doesn't matter because the trait is dominant, so that it actually, even with 1 copy, you produce enough correct hemoglobin or antithrombin. Okay. Now, it turns out we can actually use goats to produce massive amounts of the protein in a relatively short period of time. I don't know why we can't use bacteria, but they use goats. So, question, where do you think is a good place to have human antithrombin expressed in goats? Something that you can collect a lot of if you put it in the right kind of cell. Anybody like milk? Okay, so what they do is they actually take, now the goat promoter sequence is just about identical to the human promoter sequence. So the only thing that you need to do is you need to insert the actual antithrombin gene into the goat, and you do that by, in the past we did it with a, we'd inject it into the nucleus. And then that cell would divide. It was a fertilized cell, and then eventually you would get a goat that was born that produced milk that had antithrombin in it. And then you could purify it and inject it into people who had this deficiency. Oh yes, yeah, so they did it into the cell. You can kind of see this if you look closely. Just very little skinny magnified pipette. And then what you do is you actually transfer it into a mother goat and let the cells divide. It's actually, you put in an embryo, let it divide in a petri dish for a while, and then put it in the mom. Okay. And then that's what you do. So that's actually, that's human antithrombin that they're producing, and not goat antithrombin. Okay. Now, they've also been doing something else. Someone worked out the gene for fluorescence found in these jellyfish on the left. And these jellyfish are in the deep ocean where there's no light. And they use something called bioluminescence. They make this green bioluminescence. And so someone actually was able to find out which gene it was. And then they were able to use that gene to tag various proteins. And they could actually follow proteins in the protein pathway, or the constituents of a protein. So you can actually see what's going on inside of the cell now at the molecular level, because you have these tagged proteins that are tagged with a biofluorescent protein, and you can actually show up. So it's a great way to actually see it. So that's one of the things that transgenic organisms have allowed us to do. In this case, using that gene and putting it into human cells in the let's say a petri dish, and looking how they're producing various proteins, looking which genes are being turned on and by how much and turned off, all that kind of stuff. So, transgenic organisms are really common in biomedical research. Especially the biofluorescence thing has been really, really useful. We use them in research all the time in terms of biology, studying various aspects of biology. Use them in agriculture. We've genetically modified plants, engineer bacteria to produce growth hormone for cows, engineer plants to produce proteins that make them more marketable, or let's say drought tolerant, things like that. Now, it turns out that we've been looking for all sorts of ways to make cows make more milk. And it turns out there's a growth hormone, another protein called BGH, bovine, let's see, what is it? Bovine growth hormone. Yeah, okay. And that's what cows, that's the hormone that turns everything on for making milk in cows. Milk is a protein. It's a protein with, well, milk has all sorts of proteins in it. And if you give cows more BGH, they'll produce more milk. So they actually took the cow BGH gene and they put it into a bacteria. And the bacteria can serve as factories for producing BGH, and then they can take the BGH and inject it into a cow and make her make more milk. Now, which is a transgenic organism? Is the cow the transgenic organism or the bacteria? Huh? Only one. Which one has genes, new genes in it? The bacteria. They put the bacteria from the cow the gene from a cow into a bacteria. And then the bacteria produces BGH. Okay? So they've got the bacteria, it's a transgenic organism. The cow is just it's like bacteria that actually make insulin for humans. They're the transgenic organism. And when we get insulin injections, humans aren't the transgenic organism, they're just using the insulin. Okay. Okay. Now, so you can do things like that, but in terms of the take-home lesson here is I want you to think, if I ask you, which is a transgenic organism on a test question, you'll think about it and you'll say, oh, okay, it's the one with the gene from another organism. Okay. Now, I don't know if cows that have been injected with BGH actually have BGH in their milk. I don't know that at all. So I really can't say. But some people are really sketchy about using GMOs. Now, I will say that a lot of animals are given antibiotics all the time, and they do this because it apparently makes them grow faster. Makes them obviously, it kills all the bacteria that might be bad for them and all that. The thing is, it's a good way to generate resistant bacteria. And then it's a good way to actually get bacteria from, let's say, cows that have been fed bacteria, antibiotics all their lives. So there's some question about, that might not be so good. Nor might BGH be so good. But there's nothing yet that's shown up, and they've tested it, that says there's anything bad with it. Now, they are moving away from the antibiotic thing, mainly because of antibiotic-resistant bacteria. And it's a public health issue. There's been real resistance, as you can imagine, in agricultural animals, whether it's chickens or pigs or cows or whatever. In terms of making it safe for humans, they're moving away from using bacteria now. But that's not genetically modified. So this, I don't know if it's good or not. Okay. Now, I talked to you a little bit about selective breeding, artificial selection that humans have been doing for thousands of years. And this is just the teosinte. You can see, doesn't look like much of a corn. They eventually, through selective breeding, went from that to this, which is bigger kernels, and then finally to large kernels. Now, they can also, now, kernels have different constituents in them. The kind of corn that you eat is very different in taste and texture than the kind that we feed to cattle to make them grow big and lots of muscles and then slaughter them, right? So, in fact, most of the corn that we grow is that kind of corn. Or we grow corn for other parts of it that we use in cosmetics, all kinds of different things. Okay. And that was all genetically modified stuff. Well, now we can actually insert genes that affect the traits of what the corn actually produces. So the sweet corn that we eat, whether it's white or yellow, is not the kind of stuff that's actually fed to cows. And it's very little of what's actually grown in the United States compared to the total amount of corn grown. But they're now using genetically modified, in other words, they're making transgenic corn plants. One of the things they are using or putting in there is a lot of plants, obviously they get attacked by fungi, by bacteria, by viruses, and by insects. And so a lot of plants actually make their own kind of insecticide. Or they also make stuff that stops other plants from growing. So, like herbicide. So what they've done now with a lot of corn is they've genetically modified it so that they can plant it, they can spread herbicide all over the place to kill all the weeds, and it won't affect the corn plant. Whether that's good or bad, I don't know. There's always plants really make hybrids all the time. And so you can see a gene like that spreading from a modified plant to a wild other species of plant. And that would cause a real problem. As far as actually eating genetically modified corn, I don't think that's going to matter to you. You'll get the same nutrients that you would from a regular corn. Okay. Now, as an example, vitamin A is one of those vitamins that you need from your diet. And there's actually a fair amount of vitamin A deficiency in humans in various places. Africa, some parts of Asia, where the diet, they just don't get enough. Usually you get vitamin A, it's in beta-carotene. You get it from fruits and vegetables. Well, they actually took a gene that codes for beta-carotene, which is vitamin, and then they inserted it into rice, and you got this golden rice that has these different kernels, and some of the darker ones, the yellow ones, are the ones with beta-carotene in it. And so people can actually grow this, and at the same time, they can get all the beta-carotene they actually need. So it also, obviously, it can do some good. Now, there's a lot of people who think that genetically modified organisms are inherently bad. The European Union has banned them. It's really a bunch of politicians who just don't understand, and other people who don't understand. So it's actually a great technology. We often use, in the past, we used what's called DNA gun or gene gun, where we would actually shoot it into cells. But it's not a very, actually, just think of all the cells you'd have to do to make enough of a certain protein or whatever. So there's actually something that just happened. CRISPR-Cas. I'm not going to tell you what it stands for. CRISPR-Cas9. But it was first discovered, it turns out that bacteria get infected with viruses. And bacteria actually have an adaptive immune system, one that has a memory. Now, they don't have a memory like in neurons or anything like that. They have a memory in terms of what bacteria have been infected by before, which viruses. And so what this does is this assembly that they found called CRISPR, it identifies a sequence of DNA or RNA, if it's an RNA virus, and that in the past it was infected with, or that it got from its previous, that it was made from its previous bacteria when they budded off, right? So, and it recognizes the DNA or RNA from a virus. And when it does that, it then takes an enzyme called Cas9, which then cuts the enzyme, cuts the bacteria at a certain point and dices it all up and kills the virus. Okay, so it's an ingenious system for bacteria in terms of fighting off attacks by viruses. Okay, different than our immune system, but some parts are kind of the same. Well, these two women, one was a French woman who was one of the discoverers of the whole bacterial immune system with the CRISPR-Cas thing. And the other was a molecular biologist up at UC Davis. And what they did was, they thought, I wonder if we could make CRISPRs that cut genes in exactly the place we wanted to, where we could maybe cut out certain sequences of genes or put in new ones. And before this, making transgenic organisms, it was a real hit and miss thing. I mean, it sounds like, recombinant DNA, you do this. Well, oftentimes, you'd have to do it hundreds and hundreds and thousands of times just to get enough of the genes into the right place for it to actually work. It was really hit and miss. So with this, you can actually put it into exactly where you want to. And so you can put basically any gene, you can cut genes anywhere, exactly where you want, not just this hit and miss thing. You can stick in new genes. You can remove old genes. You could actually take, let's say, a human embryo, a cell, a zygote, and you could add genes, let's say, for certain traits. I mean, it's actually an incredible technology, but it's also pretty scary. So they use it, they've done it with human cells, zebra, fish cells, bacterial cells, in plants. It's an incredible technology that you probably haven't heard of, but it's going to, I really think it's going to be transformational. Whether it's good or bad, I don't know. Now, there's some very strict protocols about using it, but whether everybody will around the world, who knows? Okay. So let's say, and then there's this other thing that's going on. Clones are identical. They're like identical children, right? Or identical adults. They developed from the same egg that split in half and produced two individuals instead of one. And they share all the same genes. So that's, identical twins are like clones. In selective breeding, with a lot of trees, we did the same sort of thing. So you have these big stands that are all basically genetically similar. The problem with that is if something comes along, a new disease comes along, that has never, then these plants haven't been exposed to before, they're completely, the whole stand is susceptible instead of just some of the trees. So this has actually happened with bananas. The banana that we all eat, it's called a Cavendish, I think. There's now this new disease for bananas, and it's wiping out all the bananas. They might have to actually switch to some other banana. There's so many different varieties of bananas, and some of them taste much better than the Cavendish that you're used to. But anyway, let's say with this corn, you have all these corn plants. They're all the same. And of course, what happens if you get a new virus that attacks the corn plants? All the corn will be wiped out. So producing identical organisms that are genetically modified any way you do it, whether it's selective breeding or not, is actually not a good thing. Okay. Yeah, I think that's it for now. Good.